Good afternoon, sir.
Good afternoon, Manaswini. Okay, let's. All right. Good, good. What happened to the rest of the people? All right. So good afternoon, and we have covered a fair bit of stuff, right? We started with cloning, and we spent a lot of time on gene cloning, molecular cloning, which is also called as gene cloning, right? And yesterday we saw very briefly on reproductive cloning, another form of cloning, which is reproductive cloning, which is essentially trying to make a copy of an organism. It could be a plant, it could be an animal, right? And we saw two techniques by which this reproductive cloning could happen. One is by what is called a somatic cell nuclear transfer, where you basically take the nucleus from a somatic cell, right, and transfer it to an enucleated oocyte, right? And you give it an electric shock. Sometimes people have used certain chemical treatments, and because now this enucleated egg has 2n component, because you have taken the nucleus from a somatic cell which has 2n, deployed status, the egg behaves as though it has been fertilized, right? And it starts to divide, okay, and gives rise to the embryonic stem cells. So it's on the way to making an embryo, and embryo is made of embryonic stem cells, okay. And another technique by which you could do reproductive cloning was by embryo splitting, where you could essentially, separate these embryonic stem cells once they have grown into 4-cell, 16, 8-cell or 16-cell stage. At that point, you can treat them with certain proteases and few other chemicals. It will essentially separate these into individual cells, and these individual cells have retained the ability to grow into individual embryos. Okay, so that is what is called as embryo splitting, and this has been going on for the last 20 years. And so people have been thinking, is it possible to do a cloning for humans? And yesterday we kind of raised that question, is it okay to clone humans? Some people said yes, some people were, they've not formed their opinion. But nevertheless, in today's class, we will look at the third form of cloning, which is therapeutic cloning. Okay. The difference between reproductive cloning and therapeutic cloning is very, very thin, but very distinct. Very thin, but very distinct. Both of them would involve the use of somatic cell nuclear transfer. Now, what happens in reproductive cloning is once you have done the somatic cell nuclear transfer and you have given the electric shock or chemical treatment to that nuclei which received this, it starts divide, and then you transfer this into a surrogate mother, right? And the surrogate mother would, feed the baby and deliver at the, appropriate time, right? In therapeutic cloning, what happens is that this embryo is destroyed, and the embryonic stem cells is what is going to be used for therapeutic purposes, which means this is going to benefit a single individual or maybe a group of individuals. So that's the difference between a therapeutic cloning and the reproductive cloning. Did I make it clear? The difference between therapeutic and reproductive cloning?
Yes, sir.
Okay.
Sir, your voice was breaking on my end. Is it okay if you could repeat just the last difference part?
No, no. What I said was the in the case of therapeutic cloning, you are going to end up destroying the embryo, okay, which otherwise would have been implanted into a foster mother or a surrogate mother. In the case of therapeutic cloning, you're going to, use the cells for the benefit of a patient, okay. So that's the
Okay,
difference
sir.
between you're essentially destroying an embryo, viable embryo, right? So that's the difference between therapeutic cloning and the reproductive cloning. Okay. So today we are going to look at, very briefly. I'm going to share a screenshot. Okay. Um, so once therapeutic cloning became, an option, the US Food and Drug Administration, which kind of controls the, the marketability and the, the approval of all these clinical products, got into the picture, and it laid down certain ground rules. Okay. So this, are you able to see the screenshot?
Yes, sir, it's visible.
Yes,
Mm.
sir.
Okay. So this is from the US Food and Drug Administration, which talks about therapeutic cloning and genome modification. We'll see how these two are interlinked. Okay. So it says that the rapid advances for the past few decades in biotechnologies involving somatic cells and gene therapy offers a great potential in regenerative medicine. Okay, so this is a branch of medicine that is involved in regenerating the parts of an organ or an organ system in patients. Okay, and for the treatment of people who could be having genetic defects. For example, people who are born with cystic fibrosis, right? You could correct their cells using the genome modification techniques, and then you could derive embryonic stem cells out of those, and then you could treat the patient. So it's a long-winded, nevertheless, it's a possibility. Okay, so this SCNT, as we saw yesterday, was the one that was responsible for cloning Dolly the sheep, right? So basically, um, the somatic cells was taken from a totally different breed, okay, and it was transferred to nuclei, enucleated nuclei of a different breed of sheep, and then the eggs were given an electric shock, and then the eggs started dividing, and it formed a viable embryo. Okay, so this thing, these cells, once they are stimulated, divide into blastocyst with, these form. All of you must have studied in your embryology class, right? They become a blastula. A blastula would become a, morula, and then it'll become a gastrula. All kinds of different things. And as it commits from each step, you have some kind of a commitment that is happening, from a blastula to, to a morula to a gastrula. You have commitments that are happening. Okay, at some point in the earliest stage, they are still what is called as pluripotent. They're called. They have the ability to form any type of, tissue or organ in the body. Okay, so these are called as pluripotent stem cells. Okay, so embryonic stem cells, okay, are undifferentiated cells from an embryo that have the ability to give rise to all the tissues found in the body. As you can imagine, an embryo, a fertilized egg, which can, divide into 4 cell stage, 8 cell stage, or 16 cell stage, has this ability to give rise to entire body structure. Okay, and there has been, once the cloning became, Dolly the sheep came into picture, a lot of ethical and societal concerns were raised, okay, regarding the production of these embryonic stem cells, because as I said, for therapeutic cloning, you are essentially destroying a viable embryo. Okay, many religious groups are against killing any life form, right? Depending on which part of the world you reside, you have different regulations, different societal perceptions. Okay, so on March 7, 1997, way back when Dolly had just been created. Okay, the then President Clinton issued a memorandum and which said essentially prohibited the federal funding for cloning of human beings, or for that matter, actually, it kind of prohibited many different forms of these cloning experiments. So, although you could do certain basic studies, but when it came to therapeutics, it kind of stopped short of funding. Okay, so this explicitly prohibited federal funding for the cloning of human beings, and also it directed what is called as NBAC, or National Bioethics Advisory Commission, to thoroughly review not only the legal but the ethical issues along, for using this cloning technology. It looked at very broad things. Okay, though that it's a very voluminous report. We're not going to look at it, but in today's class we will see few of the ethical concerns. Okay, and then in 2001, that means about 4 years later, the FDA sent a letter to the entire research community asserting its regulatory authority over clinical research using these technology to create human beings, and advised that you need to have an FDA approval in order to proceed along this path. Okay, so it essentially covered everything related to this technology. Okay, and any clinical research which would involve the use of human embryos, human eggs, or enucleated stuff, you need to go through an FDA approval process. Okay, FDA approval is not granted to any process. You need to have a solid scientific rationale before FDA would approve that. Okay. Now, in the United States and in many parts of the world, this somatic cell nuclear transfer remains legal. Okay, remains legal in many parts of the world. But then in 2002, the United States federal funding, prohibited the funding for this research purpose. Okay, so that means essentially, as I said, cloning of these things, has been, although it is legal, it is not federally funded. You cannot use federal funds or government funds for doing your cloning research. But as usual in the United States, you still have several wealthy donors and several other organizations, private organizations that are willing to fund you, to explore some of these things. Okay, so one important thing where this therapeutic cloning can be useful is in 2014. See, after 2012, another decade, more than a decade passed before the FDA held a public meeting on this committee, which talks about cellular tissue and gene therapy advisory committee, to discuss oocyte modifications in assisted reproduction for the prevention of mitochondrial disease. As we all know, mitochondria is derived from the mother. Father's side has no contribution. So every mitochondria that is there in the human body is of maternal origin. It doesn't matter whether you're talking about a boy or a girl. The entire mitochondria is from the mother side, from the mother's egg. Okay. Now, when you have mutations in the mitochondria, which can be genetic, which can also be passed on to the siblings with a severe consequence. People have tried to remove the mitochondria. People have tried to do this cellular therapy to find out if it can be of use for the children. Okay, so some of these things you still need to go through the FDA. Okay, although it gives certain exceptions, it's a form of assisted reproduction. It gives some form of a leeway, but nevertheless you still have to go through the FDA's approval, okay, before you do any of these things. Okay, and as we all know, one thing that is now becoming common, as I said briefly in the opening, is that you could have somebody with a cystic fibrosis. Okay, you could take the cells from that patient, you could correct it, and then you can put it back into the patient so that the patient becomes normal. I mean, cystic fibrosis is just one example. people have taken somatic cells from the Parkinson's patients. Okay, they have made embryonic stem cells out of that somatic cell. Okay, and they've infused it back into the brains of such Parkinson's patients, and they've shown remarkable improvement. This has been done in mice. This works perfectly fine in mice. It is still being reviewed for use in human beings. Although on and off you will see some reports coming in, but as such, officially, none of the governments have approved this. Okay, so there are legalities, there are ethical issues that are associated with this thing. So I will include this, but maybe if you want, you could take a screenshot of this so that you can read this. This is something that I thought was very, very useful as a primer to understand what therapeutic cloning means and what therapeutic cloning entails. You're essentially destroying a possible embryo. Okay, so let's quickly look at maybe a couple of short these things. What are the ethics behind these therapeutic cloning? Is the screen visible?
Yes, sir, it's visible.
Yes, sir, it's visible.
Okay.
Perfect for debating in class. We're talking about the ethics of therapeutic stem cell use. So just to be clear, stem cell usage can come in two forms. You can call it reproductive.
Is it audible? Is the audio audible?
Yes, sir, it's audible.
Okay.
Cloning, or you can call it therapeutic cloning. Reproductive cloning is the obvious one. It's like making all the clones, exact replicas of yourself or your puppy or a Tyrannosaurus Rex. But for therapeutic stem cell use, we're talking about using stem cells to help cure diseases and help people by using therapeutic methods. So they're called stem cell therapies, basically. So ethics are moral principles where you have to decide if something is right or wrong. It's a very human endeavor. And it's important for scientists to consider because everything that scientists do can often have consequences that could be good and bad. So when talking about stem cells, this is a perfect place to discuss this. So we have this in the form of a flow chart here. So the main pro argument for therapeutic stem cell use is that you can decrease suffering in patients who have diseases and incurable conditions. You can help improve their lives. That's the main idea there. The con arguments, or the arguments that go against therapeutic stem cell use, depend on the source of stem cells. And nowadays, we have adult stem cells and we have embryonic stem cells. And when this stuff first came out, it was embryonic stem cells that really caused all the controversy. So actually, when we're talking about using adult stem cells, no one really has an issue with that. So it's kind of an area that people and scientists are trying to develop.
So as of now, we have talked more on embryonic stem cells, but you also have adult stem cells, okay, which are taken from the patient's cells. The patient's adult cells, which are terminally differentiated, can also be made to become like embryonic stem cells. These are called as induced pluripotent stem cells, or iPSCs, okay, which as the person said, has no controversy, except that you may have certain modifications that you may not be aware, okay. You may be taking the cells of a, say, 50-year-old person, of a patient. All those 50 years, whatever changes have happened in the DNA, in the somatic cell, you may not see the consequence, but when you use it for therapeutic purposes, we don't know what the outcome could be. So these are some of the things that are scientifically unknown. Okay.
But there are some limitations from using adult stem cells, which we're not going to discuss right here. But you'll find that out when you're researching or watching a video about the source of some of these types of cells. Stem cells are in the news all the time. You're going to find out all about that kind of stuff. Some recent Nobel Prizes have been given out regarding induced pluripotent stem cells as well too. So the con arguments, like I said, depend on the source of the stem cells. If it's adult stem cells, everyone's pretty cool with that. But if it's embryonic stem cells, then concerns get raised because we're talking about taking stem cells from humans who have not yet fully developed and have the potential to turn into new life, and then a lot of arguments will break up from here. So let's take a look a little bit further. So if it's from embryonic stem cells, some people say killing the embryo. So if you take the stem cells from this embryo, then the embryo won't be able to develop into a.
So although we say it's an embryo, in reality it is nothing but a bundle of cells which could be up to 16 cell stage. From a single fertilized egg, it could have divided into 2, 2 into 4, 4 into 8, and 8 into 16. Generally, your research would stop at that 16 cell stage. Okay. Now the question is, scientists consider the 16 cell stage as pre-embryonic. On the other hand, people from ethical and people from religious point of view, they think that once an embryo is formed, which is by the union of a sperm and egg, and once it starts dividing, they think that life is formed. Okay, so the contention is between what the ethics and what the social scientists and what the religious people think versus what a scientist thinks, right? Scientist thinks that until it acquires a form, size, and a shape, we don't consider that as life. But on the other hand, these people consider that even a single fertilized egg which has this ability to grow is called as an embryo. Okay, does that is that clear?
New human life. So killing the embryo as a result of the procedure may not be worth it. So the reason why is because to a lot of people, even if that embryo hasn't developed any nervous system or any beating heart cells or anything like that, it's still potentially a life, therefore it is a human life. Therefore killing it is like taking life away. So there are some counter arguments that exist. So some of them are exactly the opposite of what I just said. An embryo hasn't developed beating heart cells and it hasn't developed a nervous system. Whether it knows that it actually exists, that's arguable as well too. But the fact that it hasn't differentiated and turned into anything that resembles a human form or human's ability to feel pain or make decisions or think means that perhaps it's not, that big of a deal at that point. No nervous system has developed yet. We just mentioned that, so no pain. And there are other arguments as well too. The process of in vitro fertilization actually produces large numbers of embryos. So for a couple who's trying to have a baby, but they can't do it through a traditional method, and they have to get some kind of assistance to do that, a lot of extra embryos will be produced. And if they only used a few of them and they were successful and they get to have a baby, the woman becomes pregnant, what do you do with all those extra embryos? So most of the times they just get discarded, or the question.
So what do you think we should be doing with those extra embryos? Any thoughts on that?
Or can't
Adish?
we just use that to? Oh, I'm sorry. That one.
Go ahead, Manaswini. That's fine.
So can't we just use those embryos for the research purpose?
So although it says it's good to use them for research purpose, there are pro-life people who still think that nevertheless it is an embryo. For them, it's an embryo. It is life, and you're killing life. That's what they feel.
So, but I mean, okay, if you look at it that way, then technically we shouldn't be using any kind of microbes for research or experimentation, right? Because according to that definition, everything has life.
Everything is life, but for them, human life is more precious than anything else, right? So in this case, the bone of contention is human embryo. You know. Okay. Any other thought? Thank you, Manaswini. Nandini. Anybody else who wants to have a say? Vivek,
What's that?
what is your say on this? What should we be doing with those extra embryos? Okay. Am I audible? I don't see any response coming from you guys.
Is instead of discarding them or destroying them, why not just use them for some stem cell
So many people, so Manaswini, many people have decided to freeze them. They have decided to freeze them, and there are many, commercial companies that have huge liquid nitrogen canisters. And these embryos are frozen, okay, and they think that anytime, the patient, the person who generated these embryos, right? If they have a disease or if they have a problem, they could come back, grow these embryos, and then they could use it. Hopefully by then, your ethical and your morals could be changed or something like that. That's what they feel. That is something that we will be seeing in our professional ethics class. It is so unfortunate that those classes haven't started for you people. How ethics and morals can be changing under certain circumstances. We'll see some of those situations in those classes, so we will know that. Okay, so until such time, many people have decided to freeze them or to do long-term storage. For example, cord blood stem cells, are stored indefinitely. People pay money, okay, to use them for medical purpose, maybe as and when the need arises. Okay.
research. And so that is a question that comes up. So it could potentially save lives by researching with them instead of just discarding them if they're going to be thrown away anyways. And another fairly interesting argument is this idea that if those embryos were produced for the purpose of research, then that individual wouldn't have existed anyways, right? So that's kind of one of the arguments they're saying. If we actually created the embryo for the purpose of research and not for making a new human being, then that thing wouldn't have been produced anyways. So no individual that would have had the chance to live has been denied life, is what one of the counter arguments is. So this is kind of a bare bones version of the arguments that exist related to stem cells used for therapeutic purposes. And you can see most of them come from the use of embryonic stem cells as opposed to adult stem cells. So just to reiterate, one of the limitations of using adult stem cells is some of them, for example, adult blood stem cells in the bone marrow have the potential to turn into other cells, but they're limited in what they can turn into. They can only turn into other types of blood cells unless some kind of re-engineering is done. And that's what people are trying to do as well too. How do you take something that has the ability to only turn into a few dozen types of cells and trick it so that it's able to turn into other types of cells that it wasn't intended to? And so that's the idea here. But with embryonic stem cells, because they're embryonic and every one of the cells in my body came from my cells when I was an embryo, embryonic stem cells have the ability to differentiate into every single possible type of the several hundred types of cells. So the idea here is that using embryonic stem cells could yield, greater success with creating stem cell therapies. And so therefore, these are the arguments that exist with relation to this particular topic. So good thing to debate about. Whatever your own personal feeling is, I guess at least for the syllabus, you need to understand both sides. But in general, for anything that you disagree with, if you have very strong feelings about a particular morally challenging or an ethical issue, it's always good to try to really understand the counter arguments that exist, and that'll help you to solidify your own feelings, or be like a scientist, stay open-minded, and allow yourself to hear other counter arguments so that you can really trust your own feelings and decide what you think is the best way forward for science and for humanity. Go humans.
Okay. So this is another one which talks about the ethics of cloning. We'll have a quick look at this.
I think this is one question that in many ways, I think a lot of people will feel is sort of remote. Like, what does that have to do with me? Unless they have a pet that they want to clone or something like that, right? But I think it is important because it's a significant worldview issue. It has much to do with how we understand human beings
Is
as
this
made
audio
in the
audible?
image of God, and how we understand
Yes, sir.
Yes,
Audible.
sir.
Okay.
given to be stewards of what God has created. So when we talk about cloning, also referred to as somatic cell nuclear transfer, I think this is important because when people hear about cloning and are asked whether they think that we should clone human beings, the overwhelming answer is no. But when they're asked whether we should do somatic cell nuclear transfer for genetic treatment of genetic diseases, they'll say yes. Well, it's the same thing. And so when we're talking about cloning or somatic cell nuclear transfer, there's two types that I'll discuss, and both with cloning of human beings. There's some other things that we could discuss on this, but I think the cloning of human beings is the critical issue. So two types that I would discuss. One is research cloning, sometimes called therapeutic cloning, and the other is reproductive cloning. Okay, so with reproductive cloning, the purpose of this would be that it's another way for an infertile couple to have a child. In other words, it's another form of assisted reproductive technology, although I would argue it's a very different type of form, which I'll come back to. But what happens is you take the woman's egg and the nucleus is removed. It's called a enucleated egg. And then you would take a cell from, say, the father or another donor, a somatic cell. That's where somatic cell nuclear transfer comes from. But a somatic cell is a body cell. It's a non-reproductive cell that would be placed with the enucleated egg. And then it's given either a chemical treatment or an electric charge, and it fuses them together. So it acts as though fertilization has taken place. If it's successful, then it starts to divide, cell division, and you have an embryo. And so with reproductive cloning, that new embryo, just like with in vitro fertilization, that new embryo would be placed in the woman's womb for implantation. So that's reproductive cloning. Now, research cloning or therapeutic cloning is exactly the same procedure. A lot of times it's talked about as though it's completely different. It's exactly the same procedure until you have the embryo. And then what you do with research cloning is you would harvest the stem cells from that embryo, and as a result, the embryo is destroyed. The reason for doing that, much like embryonic stem cell research, in fact, it is a form of embryonic stem cell research, is that you use those stem cells, scientists would use them in research with the hope of finding cure for various diseases, or in my view, in talking to some people who are in research in this area, the idea is simply to gain greater knowledge of early human development. So how should we evaluate these procedures? My short answer to the question, is cloning ethical? My short answer is no, either form. Most people are opposed to reproductive cloning, but some will accept the advantages of research cloning and say we should go forward with that. In fact, in the United States, there's been a couple of states that have passed laws that allow for research cloning, but prohibit reproductive cloning. Now, this is interesting and tragic, because by law then, what happens is you allow the creation of a human being through cloning, but that human being must be destroyed. Imagine that. This is a law in the United States. It's not a federal law, but in certain states that allow research cloning, that requires the destruction of that human being. This ought not to happen. So the first and most basic objection to human cloning, to research cloning, is the destruction of human embryos. They're created simply to be destroyed. Now, when it comes to reproductive cloning, there are still problems with risk on human lives and destruction, because in animal trials, one might think of the great success of Dolly the sheep, which was 2 decades ago. Well, that success came only after failed attempts, well over 200, I think it was 267 failed attempts at cloning before that successful attempt. And when it comes to human beings, then are we really ready to allow for the death of so many human embryos, the destruction of embryos in clinical trials before we perfect this technique? And even if we do, the risks that are there in animal trials, again, you've had risks of deformed animals, of all kinds of things like that. Are we willing to risk those things? And we might ask ourselves, are we even in a place where we can be willing to risk those things? I mean, after all, we're not risking something on ourselves, we're risking something for someone else who has no consent in the matter, because consent can't be obtained because they haven't even been created yet. And so all of this leads me to say that even the attempt at reproductive cloning should be halted before it begins. And there is some concern that because research cloning is allowed, generally in the United States where it has been allowed, it comes with a ban on reproductive cloning. But you can imagine that if scientists are able to perfect human cloning, even if it's just research cloning, that having created those embryos, somebody will seek to use them for implantation to bring about a human being. We wouldn't know for years what potential implications that human being might bear because of our interest in cloning. So a second big concern when it comes to reproductive cloning is that in natural procreation, it is the one flesh of marriage, the husband and the wife coming together, that issues forth in the one flesh of the child. So the child is partly from its mother and partly from the father. Neither one of them completely, but here is a unique, distinct individual. And in various forms of assisted reproductive technologies, this can be maintained, but it's different with reproductive cloning. In reproductive cloning, the child who's born is an exact replica of the donor, and that donor could be the father, but that means that the father's DNA is replicated exactly. It's his DNA that is placed with the enucleated egg of the mother, and so the child that comes is essentially an identical twin. And so you think about this, that even though this child that is born is the child of the father, it's also, in terms of DNA, the twin of the father. It is this kind of thing that so disrupts the order of relationships in family and in marriage that we ought not to even attempt these things. One other consideration I would give with reproductive cloning is that some of the prospects of reproductive cloning would be to create a child who is the match for someone who needs something like bone marrow transplant or even a kidney transplant or something. So the child would be created solely for the purpose of serving the needs of another. And this is to treat that child instrumentally and to rob that person of human dignity that all human beings deserve. So even if the mother and father deeply love this child, it would remain true that the reason the child was brought into existence was solely for the purpose of supplying something that their sibling or someone else needed. So finally, and a major concern that I have with prospects of human reproductive cloning is the possibility of eugenics, and that is to engineer traits in the human race positively or eliminate defective traits. And to think that human beings have the wisdom to engineer ourselves, let alone the whole human race, is a hubris that really marks a worldview apart from God. When God gives us dominion and stewardship over what He has created, it surely doesn't include re-engineering the human race. Only God has sovereignty and wisdom to create as He will.
Thanks for watching Honest Answers. Don't forget to
All right. I hope you all like this video. Let's quickly watch another video.
So when we come to the Bible, we need to realize that it's
So why we still haven't cloned humans, right? It's not only about ethics. Okay. Let's quickly look at this video.
Clone human embryos for about 7 years. But as far as we know, no one's actually cloned a whole person. Turns out, ethics aren't the only
Is this video visible and audible?
Yes, sir.
thing holding scientists back. Cloning isn't the sci-fi marvel we think it is. It can be dangerous, often ineffective, and most of all, we just haven't thought of a good enough reason to do it. So here's why you'll probably never have to fight your evil clone. This is Dolly. Just kidding, that's a regular sheep. This is Dolly, the first mammal cloned successfully from an adult cell. She was born in 1996 after scientists figured out how to remove the DNA from the egg cell of a Scottish blackface sheep and basically replace it with the DNA of a mammary cell from a Finn Dorset sheep. They gave it a little electric shock to fuse the cell and get it replicating.
So did you see that the egg was from one particular strain of sheep, and the nucleus was from another strain of sheep?
Place the cells in the uterus of another sheep and boom, clone. This method, called reproductive cloning, could theoretically be used on humans. But this is a best case scenario. It took 277 tries for the scientists to get one Dolly. Nowadays, cloning mammals generally has a success rate of about 10 to 20%, better than 1 in 277, but still a majorly inefficient process.
Technically, it's not difficult to produce clone embryos, but human cloning has other hurdles that need to be considered.
To even research human cloning, scientists would need to ethically collect a large amount of donated eggs and find enough surrogates to carry them. But even if they made it through that logistical nightmare, the biggest issue is this.
They're going to hurt the baby, or they're going to hurt the person carrying the clone fetus.
Across the board, scientists have found that some embryos expire before they're implanted. Others result in miscarriages. And those that make it to term often die soon after birth or end up with severe abnormalities. Simply, these are risks that are easier to take when it comes to experimenting with sheep than with people. But arguably the biggest reason we haven't cloned a human being? There's not a good enough reason to. In pop culture, cloning is used to bring people back from the dead. But that's not how it works. Cloning someone would only create a twin, not a replica, since identical twins have the same genetics, but not necessarily personalities. And a never let me go scenario where organs are harvested from clones to save the rich is not only unethical, but unnecessary. Why clone an entire person when you can just make the part you need? Something theoretically therapeutic cloning can solve. Therapeutic cloning is almost identical to reproductive, except the cloned embryo is never implanted in a uterus. Instead, the embryo is cloned for the sole purpose of extracting stem cells. Stem cells have the incredible ability to turn into any other cell in the human body, which means they're great for developing new treatments for disease and have the potential to repair or regenerate tissues and organs. But no surprise, there are a lot of downsides with therapeutic cloning. The thing about stem cells is that they're a pretty limited resource. The most substantial source for embryonic stem cells, 3 to 5 day old embryos, cloned or otherwise. And when someone else's stem cells are transplanted into a patient, the body will sometimes fight them off like a disease. Some researchers believe that cloned stem cells, since they share the patient's DNA, would be less likely to be rejected. But this use case is still in the research stage. And finally, therapeutic cloning is an individualized treatment in a world where drug companies are more interested in standardized ones. And there are easier ways to create multipurpose cells nowadays, like the method for creating induced pluripotent stem cells. They're basically adult cells that have been reprogrammed to be a different type of cell.
The problem with therapeutic cloning, of course, is that you need lab personnel that is qualified to do it, specific equipment to do it. Whereas the other technique, you can just buy a kit and one person can do it in a lab that has some expertise in tissue culture.
Cloned cells still have an advantage when it comes to healthier mitochondria and the ability to grow into entire animals, whereas iPSCs often peter out. But since iPSCs safely and reliably do most everything but create entire living animals, why fund the harder, ethically ambiguous thing? So, cloning might actually have a bigger place in movies than it does in real life, because the money just isn't there. And just because we can do something doesn't mean we need to.
Your scientists were so preoccupied with whether or not they could, they didn't stop to think if they should.
So in the research for this video, I did come across one very interesting tidbit, and that is the announcement of cloned human baby Eve, who was born on December 26, 2002. And the source of this announcement is a company called Clonaid, which was formed in 1997 by the Raelian cult. And they're a cult that believes that humans were cloned from aliens, and the only way for us to reach immortality is to clone ourselves. It's been 18 years, and we haven't gotten any proof that baby Eve exists or has ever existed, but the company is still alive and well. So if any proof does come through, we will update you. Subscribe below so you don't miss it.
All right. So, quick short video on, ethics, why it has still not happened. Although, as we said yesterday, there are some reports, some reports came that, people have been cloned, but no formal announcements of that, because by and large, almost all the governments in the world had banned human cloning. Okay, so as of now, there are no reports. Although it is possible that some things might have happened and it could be hiding, and we don't know when it will come out. Okay, so any questions on therapeutic cloning?
Sir, I have a question.
Yeah, please.
So, can we do cloning like on a bigger basis? Because in the first, I think not the first, but later videos, it said that they were storing embryos. So that's not going to be economical, is it? What if the person in question never needs the embryo?
Right. These are all legal and ethical issues which have still not been addressed. For example, people say between a husband and wife, they have decided to store an embryo, in the very, very early stage. So these are tiny. And they require extremely tiny space. Okay. So you can literally store, millions of such embryos in a small liquid nitrogen canister. So that is not an issue. The issue is what do we do with these embryos once the parents don't exist, right? People who were paying for it to be kept in that frozen state, when they no longer exist, what do you do with these embryos? So that's a massive question that we are still unable to figure out. That is one thing. Did that answer your question, Nandini?
Yes, sir. Thank you.
Any other question? So it is still in the scientific realm, as we saw in the last video, although the efficiency with which these processes happen has seen a dramatic improvement, from 200 odd things to, now we have gone to, it can be done much more efficient way, but still Even this is a very, very inefficient process. Okay, and then I think our own ethics and our own morality, keeps changing. Depending on the situation, people have different stances. And so we still don't know. We still don't know if it is ethical or unethical. There are certain specific instances when what we consider as ethical, you yourself may change it, under certain situations. Okay, any other question? Anybody else? I see something in the chat box. Right. Okay, if there are no questions, then so this kind of essentially, what should I say, finishes this entire cloning part. I just wanted to give you a flavor. The main focus and applications of MolBio, this course is on gene cloning. Okay, so but I essentially wanted to give a broad view of what cloning would mean, and what all different facets of cloning are there. Okay. So we finish with this part, and then we will move on to the next other topics. All right?
Thank you, sir.
Okay, thank you.
Thank you, sir.
Okay, great.
Thank you, sir.
Thank you, sir.
Thank you, sir.